Good morning. It's May 6th, and this is your daily brief in science. Here's everything you need to know. Researchers at MIT have made a significant advancement in tuberculosis research by developing a method to label a specific glycan known as MAN-LAM within the bacterial cell wall. This glycan is crucial for studying its role in TB infection, and the new technique allows scientists to visualize its location and observe its behavior during the initial days of infection in host immune cells. The findings challenge previous assumptions that MAN-LAM was primarily shed into host cells, revealing instead that it remains attached to the bacterial cell wall for several days after infection. Tuberculosis, which infects around 10 million people annually and results in over 1 million deaths each year, is notorious for its ability to evade the host immune system. Current diagnostic methods, such as chest X-rays and sputum cultures, are often inaccessible in developing countries and have high false negative rates. This highlights the urgent need for simpler and faster diagnostic options, especially for vulnerable populations like children who may struggle to provide sputum samples. Lead researcher Laura Kiesling emphasizes the need for effective diagnostic tools, and the team aims to create a test that detects TB-associated glycans in urine. This approach would offer a faster and more affordable alternative to existing methods, utilizing the small molecule label to detect MAN-LAM in urine samples, potentially improving sensitivity in diagnosing less active cases of TB. The study successfully employed an oxaziridine tag to selectively label MAN-LAM, which enabled effective visualization and tracking of the bacteria during infection. This research underscores the importance of glycans in TB pathogenesis, as they can mislead host immune responses, complicating efforts to combat the disease. Future research will explore how this labeling technique can enhance understanding of bacterial responses to antibiotics and immune stimulation. The study received funding from prominent institutions, including the National Institutes of Health and the National Science Foundation, highlighting its significance in public health. Researchers at the Weizmann Institute of Science have made a significant discovery regarding the MTCH2 protein, commonly referred to as MITCH. Led by Professor Atan Gross, the study found that silencing MITCH in mice can enhance athletic performance and provide protection against obesity by accelerating metabolism. The research demonstrated that deleting MITCH from progenitor cells disrupts the synthesis of new fats and reduces fat accumulation. This deletion also increases cellular respiration, which leads to greater energy expenditure from fats, carbohydrates, and amino acids. In a bid to develop new treatments for obesity, Gross's lab is working on a small molecule designed to inhibit MITCH. This effort involves collaboration with Weizmann's Translational Research Unit and Yeda Research and Development Company. The study highlights a broader collaborative effort in obesity research, with contributions from institutions such as the University of Pennsylvania and the University of Texas, San Antonio. Mitochondria, known as the cell's energy producers, play a vital role in energy metabolism, and their efficiency is regulated by MITCH, which controls mitochondrial fusion. Despite the availability of innovative weight loss drugs over the past decade, these treatments often lead to a decrease in muscle mass, raising concerns about their long-term effectiveness. The urgency for effective obesity treatments is emphasized by OECD data from 2021, which indicates that 68% of adults in the United States are overweight or obese, with similar trends observed in other countries. A groundbreaking study published in Nature Communications reveals how generative artificial intelligence is transforming mitochondrial research. This innovative approach focuses on designing new mitochondrial targeting sequences, or MTSs, which are crucial for metabolic engineering and disease therapies. Lead researcher Huimin Zhao highlights the growing interest in AI applications within scientific fields, particularly in synthetic biology and biotechnology. The study demonstrates that AI-generated MTSs can significantly enhance metabolic engineering and protein delivery, offering promising therapeutic benefits. The research team utilized a deep learning framework called Variational Autoencoder to create 1 million AI-designed MTSs, experimentally validating 41 of these sequences. Remarkably, they achieved a success rate of 50 to 100% in targeting mitochondria across various cell types, including yeast, plants, and mammals. The generative AI model analyzed existing MTSs to uncover key structural features, such as positive charge and the ability to form an alpha helix, which are essential for effective mitochondrial targeting. Designing new sequences is complicated by the unique three-dimensional structural characteristics of MTSs, a challenge that generative AI effectively addresses. Currently, research is limited by a small number of characterized MTSs, typically ranging from 10 to 120 amino acids in length. This limitation often leads to the repetitive use of sequences, which can result in genetic instability during metabolic engineering due to homologous recombination. The study emphasizes the urgent need for a diverse library of MTSs. Mitochondria play a vital role in energy production and various metabolic processes, with dysfunction linked to aging and numerous diseases. Ashutosh Boob, the first author of the study, noted that the project significantly enhanced his critical thinking and scientific study design skills during his PhD. This research marks the first generative AI publication from Huimin Zhao's lab, and it underscores the extensive experimental validation conducted to support the AI findings. Overall, the study illustrates the potential of generative AI as a powerful tool in synthetic biology, potentially revolutionizing the design and application of targeting sequences in various scientific inquiries. The ASGCT 2025 annual meeting is approaching, scheduled from May 13th to May 17th in New Orleans, Louisiana. This event will feature a significant presentation from Scribe Therapeutics, which plans to showcase new data on CRISPR-based genome editing for Duchenne muscular dystrophy, or DMD. Their innovative X-editor technology will be a focal point of discussions, particularly on May 14th when they will present advancements in the novel Cas editor for precise genome editing. On May 15th, Scribe will delve into self-targeting CRISPR-Cas editor AAV vectors aimed at controllable editing in the murine central nervous system. This research is crucial, as it targets mutations associated with DMD, a severe neuromuscular disorder that can result in respiratory and cardiac failure. Scribe is collaborating with Prevail Therapeutics, a subsidiary of Eli Lilly, to present engineered self-inactivating AAV vectors utilizing their CRISPR-based XE technology. This partnership aims to demonstrate effective in vivo editing in the central nervous system of mice, representing a milestone in treating neurological and neuromuscular diseases. The results from their collaborative study are expected to showcase effective, transient, and controllable on-target genome editing within the murine CNS. In addition to their work on DMD, Scribe Therapeutics is advancing their lead candidate, STX-1150, designed to target the PCSK9 gene, which could significantly reduce LDL cholesterol levels. The company has formed strategic partnerships with major pharmaceutical firms, including Sanofi and Eli Lilly, to expedite the development of their CRISPR technologies. Scribe's overarching goal is to create safe and effective CRISPR-based genetic medicines, aiming to revolutionize treatment standards for prevalent diseases, starting with cardiometabolic conditions. Co-founded by Nobel Prize winner Jennifer Doudna, Scribe Therapeutics is supported by prominent life sciences investors and is focused on transforming the landscape of genetic medicine. A new study published in Nature raises significant concerns about the long-term effects of antibiotics on gut health. Conducted by researchers at Stanford University, the study examined the impact of a 5-day course of the antibiotic Cipro on 60 healthy adults, revealing that gut bacteria can be disrupted for up to a year following treatment. Participants in the study took 500 milligrams of Cipro daily for 5 days, with stool samples collected over a 20-week period to track changes in gut microbiome composition. The findings indicated that antibiotic use resulted in persistent antibiotic resistance in gut bacteria, lasting over 10 weeks, with some resistance detected for as long as 1 year. The research identified 513 bacterial populations that exhibited genetic mutations associated with fluoroquinolone resistance, suggesting an adaptive evolution in response to the antibiotic. This highlights the critical need for monitoring gut microbiome composition to mitigate antibiotic resistance and enhance antibiotic stewardship. Antimicrobial resistance is a growing global health issue, linked to millions of deaths each year and projected to claim nearly 40 million lives by the year 2050. The rise of antibiotic-resistant bacteria is occurring faster than the development of new antibiotics, worsening the situation. Contributing factors include the overprescribing of antibiotics, particularly in pediatric and nursing home populations. Dr. Eitan Yaffe, who led the research team, sequenced genomes of over 5,600 bacterial species and analyzed more than 2.3 million sequence variants to reach these conclusions. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.